My poster and experiment is about kinetic assay of allosteric regulation of glycogen phosphorylase B by ibuprofen. My experiment is a part of Biology 639 Advanced Biochemistry Laboratory. I was mentored by Dr. Roberto Guzman from Molecular Bioscience Department. We all know that glucose plays an important role in energy metabolism. This makes glycogen phosphorylase B a very essential enzyme as it catalyzes the conversion of glycogen to glucose and back. The enzyme is allosteric enzyme, hence its activity is affected by many compounds changing its conformation by binding to its regulatory and active sites. We investigate AMP, which activates glycogen phosphorylase allosterically, and common chemicals such as caffeine and ibuprofen. Caffeine is a very prominent compound in coffee, and ibuprofen is a commonly used medicine. The experiment reaction was opposite or reverse to the reaction of interest. As in, glucose 1-phosphate reacts with glycogen N to form inorganic phosphate and glycogen N plus 1. The reason we use the reverse of this reaction is because to monitor a reaction in kinetic assay, we need to know both the substrate concentration and the enzyme activity associated with it. Using molybdate, which when combined to inorganic phosphate forms a blue-colored reaction mixture, we can find the extent or the amount of product formed, thus the enzyme activity, by monitoring the intensity of the blue color. Here, let's look at the figure glycogen phosphorylase AMP complex. In this, the green color obviously is the glycogen phosphorylase compound. The yellow color indicates the AMP allosteric site. The blue color indicates glycogen binding site, and the red color indicates the catalytic site. Now, let's look at the results. Figure 1 shows how absorbance increases with increase in inorganic phosphate concentration in the presence of molybdate ion. It increases linearly or proportionally. In figure 2, we see how the reaction or enzyme activity can be monitored because the inorganic phosphate concentration increases with the substrate concentration. Also, this figure acts as a confirmation to inorganic phosphate being a product of the reaction. The figure 3 is of AMP activator. Now, AMP activator was expected to improve the enzyme activity, being an activator of sorts, but unfortunately, our results didn't really match to the expected results. Apart from the highest concentration of AMP, which is indicated by the inverted triangle, orange triangle. The rest of the runs with different concentrations of AMP did not show an increase in enzyme activity. We believe that further experimentation is required, but it is strongly agreed upon that the enzyme activity has to be above that of a run without AMP activator. Let's look at figure 4. Figure 4 indicates caffeine as a non-competitive inhibitor. The reason I believe that it's a non-competitive inhibitor is because the Vmax, no matter what the substrate concentration increase, never matches that of a run without the inhibitor, as you can see from the data here. This is because the non-competitive inhibitor binds to a site other than the actual site where the substrate binds. Thus, it does not compete with the substrate to inhibit an enzyme. On the other hand, ibuprofen turns out to be a competitive inhibitor, because as the substrate concentration increases, all the enzyme activity seems to match or cross that of the run without an inhibitor. It is unfortunately, data such as certain Km values and a few Vmax values do not really match the expected results, and we hope to do further experimentation would give more reliable data. I'd like to conclude by saying that the values of AMP and ibuprofen can be improved by further experimentation. I hope that this experiment forms an evidence to further inquire about ibuprofen's effect on glycogen phosphorylase B, because ibuprofen is a commonly used medicine and glycogen phosphorylase B is a very essential enzyme. Further experimentations using fluorescence spectroscopy could understand the conformation changes caused by ibuprofen on glycogen phosphorylase B, which affects its activity, and also on what groups on the compound, functional groups on the compound affects the enzyme. Thank you.